Well, Farmers Weekly's new thinking piece by Neil Wallace this week is talking about how our bees here are getting a fighting chance as scientists have identified naturally occurring compounds that induce a cleaning response among some worker bees, killing juvenile varroa mites. Now, varroa mites reproducing in brood cells, and how researchers have identified how some worker bees can open those cells, pull out the contents, and killing the mite. Well, our next guest has retired, but Emeritus Professor Alison Mercer, welcome to Serious Country, and thank you very much for all the work that you and your team have been involved in, and the University of Otago researchers on this project. How long have you all collectively been working on it?
Well, probably about 8 years or so now, so quite a long time.
And we know the varroa mite has been, a huge worry for our biosecurity to our very valuable honey industry in New Zealand. The research that has been, whereabouts has that been funded and driven through?
Both the University of Otago and through a collaboration with researchers in France. So the key researcher here was Fanny Monday, or is Fanny Monday. She's continuing this work. And Fanny was actually born in France, but she came here, first of all, to my lab at the University of Otago to do a master's degree, and then she went on to do her PhD. And during her PhD studies, she was very keen to try and answer the question, why is it that some honeybee colonies show resistance to varroa mites, whereas most colonies don't? So most colonies, once the varroa mite arrives, they usually don't survive for more than about 2 years without help from the beekeepers. So she wanted to know why some colonies are resistant. And of course, this is a question that people have been interested in all around the world. And so Fanny got this work started and made it a collaborative venture between the University of Otago and the Institute for National Agricultural Research in France.
Fantastic collaboration between the countries, and congratulations to Fanny and all of her work. So without intervention, infected honeybee colonies die within 2 years. So this particular compounds and the way that it actually, the resistance can happen, can you explain that a little bit to us in layman's terms?
Sure. Well, I'll try. So the thing is that mites are tricky. So they are able to camouflage themselves by making their bodies smell like the bodies of bees. So they're hard to detect. So bees find them hard to detect. And despite this, some colonies seem to contain worker bees that are able to detect the mites. And the ones that they can detect are the ones that are inside brood cells. So the brood cells are where the mites reproduce. And brood cells are simply cells that, instead of containing honey, the cell contains, the queen lays an egg in the cell, and the egg develops into a bee. So brood cells can contain developing bees, and the mites reproduce in those cells. And it seems that there are some worker bees that are able to detect the mites in the brood cells. And it turns out that it seems to be the juvenile mites that they detect. And any worker bee that can detect the mites will work collectively with other workers that also can detect the mites, and they open up the cells. The cells are normally capped over with a wax cap. So they open the cell, they kill the developing bee, which sounds pretty drastic, but they do this in order for the next step where they clean out the cells. And in so doing, they also clean out the mites, and so the mite reproduction is disturbed. So this, what is called varroa-sensitive hygiene behaviour, suppresses mite reproduction, and that helps the bees to cope with mites. It doesn't eradicate the mites, but it keeps the mites at a level that the colony can tolerate. So the key question here is, why do some colonies have enough workers to do this and others don't?
And so with the research, how are beekeepers being able to identify within their particular hives and to protect and make the hives more resilient going forward? How are they implementing this research into their businesses?
Well, we hope that they will be able to. At the moment, what beekeepers need to do in order to control mites and protect their colonies is they need to use miticides. And the problem with miticides is the mites rapidly become resistant to the miticides. The miticides start appearing in the honey, and miticides are expensive and not very environmentally friendly. Now, what Fanny has found is that these compounds that she's discovered are only found in brood cells that contain reproducing mites. And moreover, if she puts these compounds into cells that don't contain any mites, the workers that can detect the compounds think there are mites in the cell. So she has shown, and collaboratively, the researchers have shown, that if you inject these compounds into cells, it will trigger varroa-sensitive hygiene behaviour. So it answers two questions. What is the signal that the worker bees are using, and what is it that triggers this important hygiene behaviour? Now, because we know that, we can now hopefully design a tool where beekeepers or bee breeders are able to inject the compounds into cells. And the important thing is that the responses they induce mimic the natural varroa-sensitive hygiene behaviour. So in other words, it gives you a measure of how resistant the colonies are to the varroa mites. So if you inject these compounds into brood cells and you go away and the next day you come and have a look and all the cells have been opened up, so there's been a very strong response, you know that colony is likely to be resistant to the varroa mite. So it's a way of selecting the colonies, which we hope is going to be a very useful tool for bee breeders.
Absolutely. What work is Fanny doing in extension with Apiculture New Zealand Beekeepers Association to move this forward and to move forward with that tool? What's the sort of time frame?
Well, that's a good question. I mean, she's shown very clearly that if you use these synthesized versions of these compounds, they work. The next key thing is to find a delivery technique. So she and the people who have been helping her have been injecting individual cells, with these compounds, and that takes time. And no beekeeper or bee breeder has that as much time. So what they're working on now is some delivery tool that would be robust, reliable, and inexpensive, because if we can produce a delivery tool like that, then that would be a great benefit to beekeepers and bee breeders alike.
What a fantastic piece of research. Thank you so much, Emeritus Professor Alison Mercer of Otago University's Department of Zoology, speaking there on behalf of Fanny Mondo's work as a doctoral candidate, working in there with the French as well, French researchers. A great combination collaboration between France and New Zealand to work on what is a global issue with varroa mite. Now, after the break, we're joined by Dairy Environment Leaders Chair Melissa Slattery from Wellington, from the Dairy Environment Leaders Annual Forum, to talk about how they are rising to meet environmental challenges and support each other through this change. We're going to get an update from Melissa on the thoughts that have come out of the rooms of Wellington.
Oh, nice boots.
Yeah, thanks. They're new.
Ben, you alright?
Oh, yeah, sorry, mate. Just meerkatting pretty hard, eh? Yeah. I just said to myself, Ben, work hard, deserve new boots.